Yeah, great. Thanks so much for the invitation. So just to give you a brief overview, we're going to split the talk in half, and I'll be giving you some context and background, and then Catlin is going to talk about an actual bat study that we ran out of China and a little bit about the types of data that we got for that. Okay. So just to give you a little bit of an overview of generally what my lab works on is we're interested in understanding the motion of immune cells. I like to think of immune responses as a complex dance of cells that is tightly regulated, and the motion of immune cells is really a critical factor in this process. So of course, immune cells have to actually arrive at sites of infection to be able to help clear them. And so as a lab, generally our research focuses on a number of different questions around understanding immune cell migratory processes and how they organize effective immunity. We have some work trying to understand how T cells responses go wrong when immune cell migration is defective, and also projects around trying to understand what cell extrinsic and intrinsic factors regulate T cell migration. So I'm not really going to go into that at all much more. If you're interested, you can check out our website. Really what I wanted to talk about today is some parallel work that we've been developing to look at reservoir host immune responses to emerging zoonotic viruses, with a particular emphasis on trying to understand bat immunology. And as I'll talk about a little bit later on, this is work that started back when I was a PhD student in 2002. And over the last 10 years or so, I've been trying to develop a bat immunology program, in part in help and collaboration with Rolf Müller. So to set the scene, although we're probably all experts by now in emerging infectious diseases, I wanted to talk a little bit about what they are and some patterns that are important here. So if we take the broadest possible definition, emerging infectious diseases, of course, includes things that have recently evolved and entered the human population for the first time. But in the broadest sense, it also includes pathogens that are increasing in incidence or expanding in geographic range, or that have significantly altered their pathological or clinical presentation. So in this broadest definition, it also includes bacteria that have acquired antibiotic resistance. And if you plot this out from the 1940s or so, the cases of emergence, as Peter Daszak's group did here in this Nature paper, what you can appreciate on the one hand is that a lot of these emerging infectious diseases are zoonotic in origin, which is the white, red, and orange component of this bar, about 60 to 75%. And also that the frequency of emergence is increasing over time, from the time that this particular study looked back to the 1940s or so. So it's been steadily increasing. There's this peak in the 1980s that is accounted for by the emergence of HIV in humans. And of course, as a result, a number of opportunistic infections that hadn't been observed in humans before. So hence this peak here. But the overall trend holds to this day. And of course, as we're experiencing now, there's a very great potential for the rapid dissemination of these emerging infections across the globe. So, of course, these things are inherently unpredictable, but there are patterns that exist. For instance, there have been geographical hotspots described where emergence is more likely of these novel pathogens. And this tends to happen where there's both high biodiversity as well as high human population density. One place where this is the case is in China. And important to note is that nearly all recent pandemics have a viral etiology with animal origins. So viruses account for a big fraction of emerging infectious diseases. This is a curve that shows sort of the cumulative count of viral species that have been reported to infect humans, starting with the first human virus that was identified in 1901, which is yellow fever virus. Up to about 2018, there were about 263 virus species from 25 different viral families that were described to infect humans. But this is really only the tip of the iceberg. It's been estimated in a study that was published in 2013 that there's something like 1.7 million undiscovered viruses out there, of which something like 600,000 to 800,000 might have zoonotic potential. So emergence of viruses into the human population is something that's going to keep happening. And just to mention a few by example, using this map. So of course, I talked about HIV very briefly, and I'll come back to HIV. There's dengue virus, West Nile virus, yellow fever virus, various influenza strains, Nipah and Hendra virus that I'll also get back to a little bit later, and Ebola virus. So there are a lot of different viruses, including Zika most recently, that's not on this map yet because this was published in 2014, that have had an enormous impact in human population, not to mention the reason that we're all sitting at home and not interacting in person right now, which is Sars Coronavirus 2. And one of the things that I think the current pandemic is amply demonstrating is that there are very few treatment options that we have when such new viruses cross into the human population. Vaccines take quite a while to develop, and if we're lucky, there might be some antivirals that work against a particular new virus. Otherwise, we're left with using convalescent serum in the cases of very severe disease. But overall, there's really not much we can do, and yet these viruses have enormous economic and health burdens, as we're seeing right now. So why are these viruses so pathogenic, and is there anything that we can do to develop more broadly applicable treatment methods? What do we know about the pathogenesis of non-natural viruses in non-natural human hosts? So of course, one of the things that's very clear is that these cross-species jumps can result in severe and sometimes even fatal disease. And this is a phase diagram that we made in a publication where we were speculating about some of the patterns regarding pathogenesis in both natural and non-natural hosts for these viruses in a review that was published in Cell a while back. And basically, just to illustrate that if you plot pathogen burden against deviation from health, then you can think of at least 4 different outcomes. One is that there's death of the novel host because of uncontrolled viral replication. More likely, there might be an outcome between 2 and 3, where there's an immune response that's ultimately ineffective and there's loss of health up to death, versus viral clearance at a cost of some loss of health. And also in some rare instances, you might get persistent infection, as is the case, of course, in HIV, but also in some instances of Ebola virus infection where Ebola was found to persist in immunoprivileged sites like the eye or the testis. So one of the very interesting things across different viruses that have made this host species jump is that oftentimes, in fact, the host response is playing an important role in contributing to disease pathogenesis. And I'm just going to illustrate this with 2 examples, one being SARS, where it was very clear from studies that were performed back in 2003 or so, that there's early dysregulated innate responses that are playing an important role in local tissue damage in the lung. And one of the things that was shown is that poor outcome is associated with persistent type 1 interferon production that's ultimately also leads to impaired adaptive immunity. And this is something that's also been seen in COVID-19, where some early innate immune responses, such as cytokine production of IL-6 or IL-1 beta, are associated with severity of disease. The immune response also plays an important role in hemorrhagic fever pathogenesis. So there are inflammatory mediators that are released that are important in vascular leakage, and the activation of the coagulation cascades. You also see this very striking dip in lymphocyte counts that you actually also see in COVID-19. And ultimately, in the worst case scenario, this leads to the vascular leakage, leads to organ failure and septic shock-like syndrome. So to make the point that really for a lot of these pathogenic viruses in humans, the immune response is an important contributor to disease. And this brings me to an important concept that is fairly new in mammalian immunology, and that was first introduced by David Schneider in a perspective that he wrote back in 2008, where he postulated that there are really two distinct host defense strategies upon infection with a pathogen, the first being resistance to infection. And this tends to be the focus of immunologists thus far, how the immune system actively restricts pathogen replication in order to reduce pathogen burden. So this is illustrated here. If there's some distribution in a population in terms of deviation from health with respect to pathogen load, then greater resistance just means that the average pathogen burden is lower. And this is, of course, the type of mechanism that intervention strategies have focused on the most in terms of generating vaccines, where the goal is really to restrict pathogen replication very early on, but also in the development of antivirals. So there, the primary emphasis is to study the underlying mechanisms of disease and how the immune system actively restricts pathogen replication. But really what was new that David Schneider proposed is that there's this alternative strategy that he termed disease tolerance, where rather than limiting the pathogen replication, what the ultimate goal is to limit the fitness costs that are associated with infection by the pathogen. And these fitness costs might be a result of pathogen replication directly or a result of the immune responses. And so that here the idea is that really what is being affected is the amount of deviation from health with greater pathogen load. And so that this blue line illustrates a population that has a higher tolerance for infection with a particular pathogen. And we really know very little about what tolerance mechanisms are at play that can limit damage that is resulting from infection. And so one of the things that we proposed in this review in Cell was that maybe one of the commonalities amongst different reservoir hosts is that as a result of their longer evolutionary relationship with a particular virus that they carry, this has led to them evolving a greater resilience to disease. And what we proposed is that this is at least in part due to differences in the quality, the kinetics, or the magnitude of the immune response that is elicited. And one important point here too is that this disease tolerance or disease resilient is really pathogen specific. So if that reservoir host encounters another pathogen that is novel to that particular host species, then this disease resilience may not apply in that case either. So why are we interested in studying reservoir host immunology? So one argument is that really in order to understand the transmission dynamics and the likelihood of a virus spilling over into the human population requires some understanding of reservoir host immunity. So to be able to work out whether or not infection is acute or persistent, whether there's memory developed and as a result of encounter the reservoir host to the infection, if there are any kind of seasonal or environmental effects on immunity. And knowing these things will give us a better understanding of whether or not this particular virus is likely to make the species jump. On the other hand, investigating the mechanisms that underlie this greater resilience in reservoir hosts may also inform new approaches for treatments in humans. So the idea is that this disease resilience is an active process, and if we can understand better what allows a reservoir host to be resilient to infection with a pathogenic virus, then maybe that's something that allows us to modulate the immune response in humans in order to achieve the same resilience. And of course, it's also a rich opportunity for elucidating fundamental immunological processes in other species that are maybe not the traditional focus of immunological studies, like mice, which are our favorite model, or humans. What do we know about reservoir host immunology? Well, the short answer is that we don't know a lot. This is the table that we made back in 2015, but I think it's largely still true, is that for most of these viruses that have made the species jump, where we do know what the natural host is, we really don't know much about the immune response beyond whether or not there's seroconversion in the reservoir hosts. One particular reservoir host that has been more studied with regard to the immune response has been the African primate species infected with a simian immunodeficiency virus, which are the reservoir hosts and the zoonotic origin of human immunodeficiency virus. And this is actually how I initially got into this topic, because as a PhD student, I was studying the immune responses of pseudomangabes, which are a natural primate reservoir for SIVs that actually were the zoonotic origin of HIV-2 in humans. And the rhesus macaques, which are an Asian monkey that develop AIDS-like symptoms, just like humans infected with HIV. And so as a PhD student, I was studying immune responses of these two primate species upon infection, experimental infection with pseudomangabe SIV strain. And what was interesting is that in both of these primate species, you get very high and chronic viral replication, but the outcome, the disease outcome, is very different. So pseudomangabes don't get sick and rhesus macaques develop AIDS. And one of the things that I described in the work that I did as a PhD student was really that the early innate recognition and subsequent innate immune response that the pseudomangabes were raising to SIV was fundamentally different from that of rhesus macaques. And that probably has an important consequence for whether or not they develop disease. And in particular, we characterize the type 1 interferon response being important here, and difference in magnitude between pseudomangabes and rhesus macaques. And just to say that this is something that was subsequently also shown in African green monkeys, which are also reservoir hosts for SIVs, but not ones that are made the zoonotic jump to humans. So just to show, this is a transcriptional analysis looking at magnitude of change in type 1 interferon response genes. And what you can see is that there's only a very transient increase in type 1 interferon response genes that goes away over time. And in the rhesus macaques, this is something that is maintained and stays high over long periods of time. So what I started wondering back then was whether there are any generalizable lessons from studying reservoir hosts in order to better understand mechanisms underlying disease tolerance. And I became very interested in bats as a reservoir host for RNA viruses because it was clear that they were reservoirs for various different viruses. So of course, it's been known for a long time that they harbor and transmit rabies virus, but they had also been described to be the reservoir hosts for filoviruses such as Ebola and Marburg, Hendra and Nipah viruses, and coronaviruses such as SARS and MERS at the time. And interestingly, subsequent sequencing of their virums also uncovered viruses ancestral to viruses that circulate in the human population, such as measles, mumps, hepatitis B, and hepatitis C. And one of the interesting patterns when you look at the genomic material of these different viruses for which bats are reservoirs is that they all seem to be single-stranded RNA viruses, with the exception of hepatitis B that actually also replicates via single-stranded RNA intermediate. And I'll get back to why this is interesting and maybe important. So this raises the question of whether bats are special. One thing to note is that they're also susceptible to diseases upon infection with particular pathogens. In some instances, the disease being a result of dysregulated and damaging immune responses. So one example here is the psychophilic fungus P. destructans, which infects hibernating North American bats in the ears, their wings, and their muzzles. And that can be very damaging when they return to euthermia, and also can result in them coming out of hibernation early with various energy expenditure costs. So they're not special in the sense that they're resistant or tolerant to all infections. Another important thing to point out is that Chiroptera are a diverse mammalian order with more than 1,300 different species. So one bat species may not necessarily be the same as another bat species with regard to the immune responses that they raise or their specific susceptibilities to different pathogens. Whether or not there's something about their physiological or ecological traits that make them more likely to maintain zoonotic viruses or transmit them to humans is something that still remains quite controversial. So there was a paper that was published back in 2017 by Peter Daszak's group that showed that bats are disproportionately harboring viruses that have zoonotic potential, while not having any increase in total viral richness compared to other mammalian orders. However, more recently, there was a paper that was published by Daniel Streicher in PNAS just a month or so ago, where they showed that probably the variation in the number of zoonoses from different mammalian orders largely comes as a result of the number of virus species that they harbor, and so that zoonotic potential is largely host neutral. And it's just that species that are more rich reservoir groups just have more virus species and thus a larger number of zoonotic species. And so one of the things that they suggest in this paper is that, in fact, the relationship that was found by Peter Daszak between bats and zoonotic viruses in this previous study was more a function of the particular virus families that are harbored by bats, because some virus families are just more likely to have zoonotic potential than others, and whether or not these particular viruses are harbored by bats or other mammals doesn't really affect that likelihood of transmitting to humans. So basically what they showed is that the zoonotic ability is more a feature of the virus itself than of the host association that it has. But the data that we really have so far is not granular enough that we can answer questions about whether or not viruses from different reservoirs differ in their pathogenicity or in their ability to transmit between humans or being able to spill over into the human population. So we just don't have the data to be able to answer that question. What does come out of the comprehensive meta-analysis that Peter Daszak's group did is that there are certain patterns in host-virus relationships. So one of the interesting things that came out of this analysis that is shown more quantitatively here that I already kind of alluded to earlier on is that bats as a group have a much greater richness in viruses that are single-stranded RNA viruses, such as Bunya virus, rhabdovirus, or flaviviruses, shown here, the darker red, compared to the primates that are very rich in DNA viruses, such as herpes viruses. So there might be fundamental differences between different mammalian species in terms of the viral classes that they harbor. And this is interesting because the immune system also recognizes viruses by their genomic material or their genomic intermediates. And that's a feature that's shared by different viral families. And so that's summarized here. So the immune system distinguishes viruses in part by innate sensors, which detect viral nucleic acids, and the innate immune cells express either in the cytosol or on the cell surface different pattern recognition receptors, some of which that I list here, that recognize viruses by the genomic material that they have. And so we classify viruses according to the Baltimore classification scheme, which are these numbers here that are based on what the genomic material is that the virus uses to replicate. So for instance, DNA viruses are recognized by the AIM2-like cytosolic DNA sensors as compared to the single-stranded positive sense or negative sense RNA viruses that are recognized, for instance, by toll-like receptor 7 or RIG-I-like receptors. Whether or not bats are unique hosts of zoonotic viruses, they're certainly unique mammals in many ways. They're the only mammals that are capable of sustained powered flight. They have exceptionally long lifespans despite having very small body sizes and high metabolic rates. They live in extremely large colonies. Some bat species hibernate. Some bat species can use echolocation. And so the question that we're really interested in asking, whether there are unique life history traits that we as humans have that make us particularly susceptible to generating damaging immune responses to RNA viruses, or conversely, whether the physiology of bat makes them particularly resilient to infection. There are a lot of challenges in studying bat immunology. We as immunologists, we love using flow cytometry, which is reliant on having monoclonal antibodies, which for many bat markers and cytokines, we just don't have available, although these are starting to be developed. Another challenge is really being able to establish natural infection studies. Of course, this involves importing specific bat species with all the hoops that involves regarding quarantining them, making sure they're pathogen-free. Then you need specific biosafety facilities to infect them experimentally. Oftentimes, it's quite difficult to figure out what the virus inoculum should be, the dose or the route of infection, because we simply don't know how they're naturally getting infected, and this can greatly impact whether or not the infection actually takes and whether it takes its natural course. And then a question is also, what do we compare this natural infection with? Is it humans? Is it another animal model that does develop disease? So far, there are a number of studies that have been done. Primarily, they focused on two bat species, the Pteropus alecto, which are the Australian black flying fox that have been shown to harbor both Hendra and Nipah viruses, and then the Egyptian Rosetta species that have been demonstrated to harbor Marburg virus and may also be the reservoir for Ebola. And primarily the studies that have been done have taken one of three approaches. They've either been comparative genomic studies, they've been in vitro cell culture assays looking at whether or not the virus replicates in different cells, immortalized cell lines, or experimental infection studies, where so far the primary emphasis has been on figuring out whether there's any kind of detrimental health outcome or whether there's a viral replication. So very briefly, to summarize some of what has been described. So the comparative genomic studies, the primary results from those types of studies are to show which genes are present or absent in different bat lineages, what genes are positively selected. One interesting class that has been shown to be positively selected are DNA damage repair genes. And it's been hypothesized that might have evolved as a result of avoiding the detrimental effects of reactive oxidant species production during very metabolically costly flight. For instance, the loss of the Payan gene family, such as AIM2, which is an important cytosolic sensor of DNA that I mentioned earlier. But there are also lineage-specific, species-specific findings that are sometimes go in opposite directions. For instance, in the Therapus, there's been a loss of NK cell receptors versus Rosetta species, an expansion, also in the type 1 interferon locus that has contracted in the Therapus species. And then there's been different innate immune pathways that have been investigated in cell culture to show that they might be fundamentally different. So there are a lot of intriguing things from these studies that suggest that there might be differences in immune responses in bats. And experimental infection studies have shown very nicely in studies run by Jonathan Towner at the CDC in the US have shown that infection of the Rosetta species with Marburg virus results in viral replication and shedding through oral secretions, feces, and urine without any signs of morbidity. So to lead into Catlin's part where she takes over, really what we would like to do is to capture both the magnitude and the kinetics as well as the quality of the very early innate response of bat primary immune cells. And in particular study antiviral responses in a wild bat species of interest, and then be able to compare this to responses that humans make. And sort of as part of that bigger picture that we want to develop is to also comprehensively survey the virome of the bat population that we're sampling to study their immune responses, to establish some of the baseline parameters of circulating immune cell populations and how those differ in humans using single cell sequencing approaches, to investigate the in vivo responses of naturally infected bats, as well as determine measures of possible impact that infection might have on bats using sensitive measures. For instance, some of the tools that Rolf Muller has been developing looking at how bats fly and whether there are any differences or not if they are infected. And I will let Catlin take over from here.
Thank you very much, Judith. That was great. While Catlin takes over, if there's immediate short questions for Judith, maybe we can take one or two, but it should be immediately on the material. I will have a final discussion once Catlin is through. Anybody who cannot wait to ask a question? Okay, if that's not the case, then we just continue with Catlin and reserve the discussion for when we have the complete picture.
Okay, thanks, Rolf. Yeah, so to get into the actual work that we've done so far, addressing some of those goals that Judith mentioned, I just want to put the project back into context of this idea she mentioned earlier about health as an active process and the question of what factors contribute to resilience to infection or disease. And one thing we know that can influence the disease course is the early innate response. So your innate immune system is the first line of defense against foreign invaders. And that can really set the tone for the potential for disease. So these pathogens, whether they're fungi, bacteria, viruses, protozoa, they have these conserved microbial components that we call pathogen-associated molecular patterns, or PAMPs. And these PAMPs are sensed through specialized receptors called pattern recognition receptors, or PRRs, that Judith mentioned briefly. And the identification of the PAMPs by these PRRs can initiate the immune response. And these PRRs are expressed throughout the cell, on the cell surface, in the cytosol, in the endosomal compartments, and so forth. And as she mentioned, there are multiple classes of PRRs. And for the purpose of our project, I'll just focus on toll-like receptors, or TLRs. And as Judith mentioned, there's a whole family of TLRs. And they're differentiated by those microbial components that they recognize. And so this is nice because depending on what you're infected with, a specific TLR will recognize that PAMP, and that will affect the kind of immune response that you have. So thinking about bats as reservoir hosts for viruses in particular, the viruses that are disease-causing in humans, we want to know what exactly that early innate immune response looks like when a bat encounters a new pathogen, given that that innate immune response can set the tone for disease or lack thereof. So for our pilot study, we were able to piggyback onto the second year of a much larger project funded by DARPA called SPIRIT, the Species species-inspired research for innovative treatments led by Shaw Warren out of Boston at Mass Gen Hospital. And basically the goal of SPIRIT is to compare the immune responses of a variety of different species to sepsis. And they do this by drawing blood from the organism and then stimulating the circulating white blood cells that are in that blood with different TLR ligands mimicking different infections, and then evaluating the gene expression of those immune cells. So in the first year of the SPIRIT project, these were the species that they tested, 11 species in total. And you can see that, importantly, they used species that were both susceptible to sepsis and more tolerant to sepsis, because again, the idea here is, what does that early innate immune response look like between these two groups and what's different? And what can that tell us then about resilience to infection? So yeah, when we wanted to join in the second year, we wanted to use bats. But as Judith mentioned, not all bats are created equally. So we needed to decide what species we wanted to focus on. So we've, for many years now, actually been interested in coronavirus. The data I'm going to show you comes from actually work that we did in 2017, so well before the pandemic. But just to give you a little bit of background, coronaviruses can be split into 4 subcategories: alpha, beta, gamma, and delta. I'm only showing alpha and beta coronaviruses here because of the coronaviruses that are endemic to humans, they all fall in these 2 genera. So besides the SARS coronaviruses that have been really high profile in the last couple decades, there are actually 4 other coronaviruses that are endemic to humans. So 2 of those are alpha coronaviruses, NL63 and 229E. And the most similar subspecies of coronaviruses that have been identified in non-human animals are this group here. So 3 of them were actually identified in 2 different genera of bats, and one of them, porcine epidemic diarrhea virus, was isolated from pigs. The other 2 are beta coronaviruses, OC43 and HKU1. And the most similar subspecies identified to these outside of humans was this murine hepatitis virus, or MHV. But the reason you probably haven't heard a whole lot about these coronaviruses is because the disease that they cause in humans is not as severe as the disease caused by the SARS coronaviruses. So that's what we were really interested in. So the SARS coronaviruses highlighted in blue are the causative agents of the 2003 SARS epidemic, and that's really what we wanted to focus on. So the 2 subspecies most similar to those that were identified outside of humans were identified in Rhinolaphus sinicus. And so, and also there were other reports of it being found in Hippocyterus species. So for those reasons, we really wanted to focus on these 2 species. And of course, since then, SARS-CoV-2 has hit the scene. And very recently, the most similar subspecies identified was also identified in Rhinolaphus finis. So we really wanted to focus on these 2 genera of bats. Ultimately, at the end, we decided to go with Hippocyterus, in particular Hippocyterus pratii, for a number of reasons. So aside from that, as Judith mentioned earlier, there were ongoing studies in Pteropus and Rosetta species, so we wanted to do something different. But we went with Hippocyterus because, as I mentioned, we would have to draw blood from the organisms. And obviously the size of the organism dictates how much blood you can draw, so that's a very limiting factor for us. And Rhinolaphus bats as a whole are quite small, so we would really not be able to draw enough blood to do the kind of studies that we wanted to. And for the Rhinolaphus species that were large enough, we didn't have access to them. So we focused on the next closest relative, Hippocyterus, and in particular the pratii, because Rolf's lab had a really nice map of the different caves that they had identified as roosting sites for the Hippocyterus pratii. I should note that although the pratii are larger, the blood volume still remained a large challenge that I'll talk about later on. So ahead of going to China, we prepared the immunological stimuli that we would use. So again, SPIRIT was really interested in sepsis, so they primarily used LPS. LPS is a component of the cell wall of gram-negative bacteria, and it acts through TLR4. And so we used 3 different concentrations of LPS, 10, 100, and 1,000 nanograms per ml. But because we're also interested in bats as viral reservoir hosts in particular, we also used poly-IC, which acts through TLR3. So we began preparing all of those tubes spiked with those different stimuli, but in addition, we also had baseline control tubes. So this would be just media without any stimuli for each bat, because we, of course, wanted to compare the stimulated to the unstimulated cells. And then we also had 3 time points, 2 hours, 6 hours, and 24 hours of stimulation, because as Judith mentioned, not only is the magnitude, the strength of the immune response important, but also the kinetics. How long does it take? How quickly does the response come up? So we shipped those off to China, and then this is just the different sites where the project actually took place. So primarily at Shandong University, where Rolf's lab was at the time. So this is where the bats were actually housed. This is where I did the blood draws and the stimulations. And then you can see this is the route that we took driving down to southern China where the bats actually roost. So our first collection site was in Jiangxi province. This was an abandoned building that actually wasn't on the cave map, but we started sort of pulling off the road and looking at any building that we passed, knowing that bats will kind of roost anywhere they can find. And so we did stumble upon this pretty good sized colony. And then we moved on to Fujian province and found this very large cave, which was our second collection site. And on the way, we did visit a couple other caves and as well as a reservoir on the coast, but the colonies were either too small to capture from or they had moved on from that roosting site. So this is, let me turn off the audio because the bats are very loud, but this is just an example of kind of colony size we might encounter, as well as the caves where they roost. So you can see there are probably at least 1,000 bats in this colony, and the cave ceiling was very high where they roosted, so this actually presented an unforeseen physical roadblock that we did manage to get over somewhat gracelessly. But this is what the transport and housing looked like. So we had these plastic aquaria that we had set up with this corrugated cardboard that we shredded to create little perches for them. And then we kept them hydrated throughout the journey, of course, and because it was the dead of summer, it was about 45 degrees Celsius at the sample sites. So we hydrated them pretty regularly just by sort of squirting them with water and allowing it to evaporate so they could stay cool. And then this is back at Shandong University. This is the Rolf's aviary. So there are 3 different runs here where the bats could fly around. And there are perches on the walls and little places for them to hide. And then these are the mealworms that they were fed while they were in captivity. So as far as the actual blood draws go, this is me here. This is one of Rolf's students restraining the bat for me. We basically set up these absorbent pads over these towels to create a cushioned sort of elevated surface for them. And this allowed me to angle the wing down so that the blood flow would kind of increase a little bit. And I didn't do the blood draws in maybe the traditional way you might think, where you put a needle in and you pull back the plunger, because that kind of vacuum can actually collapse the blood vessels, and I didn't want to create any long-lasting damage. So basically I just created a small puncture site with a 28 gauge needle, and then I used these heparinized capillaries to draw the blood out that way. Another thing I had to do before the draws was to sort of gently rub the wings to encourage greater circulation, because in the aviaries they weren't flying around as much, which really reduces their circulation. And I did the draws from the ulnar artery here. You can see running right below the bone. And then after the draw, I would just apply pressure with gauze to make sure that the bleeding had stopped and make sure that the wings opening and closing didn't reopen the puncture site. So you can see that there, that the bleeding has stopped. So he's not very happy. But again, it goes back to the amount of blood drawn. So before doing the blood draws, I weighed each bat so I could calculate the maximal amount of blood I could draw that would still save the bat. And going into this project, we estimated, based on the average size of this species, that we would be able to draw safely around 200 microliters per bat. And so based on this blood volume and based on trials that I did on mice back at McGill before we did this project, we figured that 50 microliters per stimulation would give us enough material to work with. So at 200 microliters per bat, that's 4 different conditions. So this is the matrix that we came up with. So again, for each bat, one of those conditions has to be the unstimulated control, which left us enough volume for 3 other stim conditions. So in the end, we calculated that we would need 20 bats in order to repeat each stimulation condition 5 independent times. And we used a random number generator to basically assign each bat to a condition. Now you can see here in red some instances where I wasn't able to get enough blood, and so I had to kind of shuffle around with samples where I could get more blood, but in the end we were able to do each condition 5 times. So what the actual stims looked like is pretty simple. So as I mentioned, we prepared the tubes ahead of time and shipped them over. And so really it was just a matter of adding the 50 microliters of blood to the stim tubes and incubating them for the allotted time at 37 degrees Celsius. And then once that was done, I just spun them down and collected the serum and stored that separately at -80 degrees Celsius, which left just the white blood cells and the red blood cells. So I lysed the red blood cells, leaving only the white blood cells, which I stored in an RNA preservation buffer at -80. So now the samples are ready to go to get back to Canada, but the whole export-import process was a whole other thing that we dealt with. So ahead of going to China, we had to get an animal ethics permit from McGill, because even if you don't perform your work at McGill, anything you do with animals has to be approved. So we got a permit for the capture and housing of wild bats, as well as the blood sampling. We got an import permit for the biological samples from the Canada Food Inspection Agency. One of the roadblocks was getting an export permit, because China actually has very strict prohibitions on when you can export, and biological samples is one of them, unless you get a special permit. So we worked with this company, thanks to Rolf and Weijing, this company DGM, that can issue such a permit. So basically you have to send them part of your sample and they can test it and make sure it's not hazardous, and then issue you a permit, and only then can you arrange for shipping. Another issue is that we had to keep our samples frozen, but dry ice is actually not permitted in China for shipping. So we use these Credo cubes, which you can freeze, and they will retain that temperature for up to 5 days. So we were able to keep our samples at -80 for the duration of the shipping. And finally, we got the samples back to Canada. We got them all organized and barcoded, and then we sent them off to the US, where they actually did the extraction and RNA sequencing. And then they sent us back the reads to analyze, the gene list to analyze. So all of this analysis was done by Dakota Rogers. He's actually on this call, another grad student in Judith's lab. But what I'm going to show you is just the LPS data, because we only had LPS human data to compare with at the time. So for those of you who aren't familiar, this is called a principal component analysis. Basically, it's a reduction of your data to the two factors that account for the most variation in your samples. So dimension 1 accounts for the most variation. So if you look at humans, you can see that the clustering of the samples is accounted for by these are the media alone groups, and these are the LPS stimulated groups, which means that the majority of the variation was caused by whether or not they were stimulated with LPS, which makes sense. And then in the Y direction, what separates out here is actually the incubation time. Now, right off the bat, you can see, no pun intended, but you can see that the bat samples are actually not nearly as uniform as the humans, and there are a number of factors that can contribute to that. So these were wild bats that we were capturing. We weren't able to control for age, whether or not they were infected. In addition, the stress of transport and housing, although we did give them a few days to acclimate to their new home, can definitely influence the immune response. And on top of that, these human samples, you can get enough blood from humans to do each stimulation condition. So in that way, you're able to eliminate interhuman variability. But despite all of those factors, what we can see is that as you begin increasing the dose of LPS, clusters do start to fall out in spite of that. And what's perhaps more clear here, what Dakota is showing here, is that there's a difference in the magnitude and the kinetics of the response to LPS. So if we look at bats, in the 2-hour stimulation time, there are no differentially expressed genes that come up in this short time period. You start to see some genes come up around 6 hours and a little bit more by 24 hours. But when we compare to humans, what's really striking here is that even at just 4 hours of incubation, you see thousands of differentially expressed genes coming up. So these cells are having a very strong response. And at 24 hours, even though it has gone down, we're still in the thousands. So not only is the gene expression later, coming up later in bats, but the magnitude of that response is also dampened compared to the humans. And so if we look here at the 24-hour time point, the 1,000 nanograms per ml LPS concentration, and we look at what genes fall into those categories and what's similar, you can see that 190 genes come up in both bats and humans in this time point. And what I want to highlight here are just a few of those genes as an example. And these genes that I'm highlighting are actually downregulated in bats. And those are IL-6, GCSF, and CXCL8. So these are cytokines and chemokines that are really important in the inflammatory process. And IL-6, actually, Judith mentioned earlier. And it's particularly interesting because, so for humans who are COVID-19 positive who have very severe symptoms, IL-6 has been proposed as a therapeutic target. So inhibiting IL-6 in order to attenuate that inflammation and try and dampen the immunopathology. So that was really interesting for us to see, knowing that now, given this data that we had a few years ago. So with that I'll just end on the take-home messages. The first is, of course, that bats are clearly an important reservoir of viruses that are disease-causing in humans. And then, as Judith mentioned, spillover events will continue to occur. I think we know that very well right now, and that each time that happens, waiting for a vaccine is just not a good option because it can take ages for that to happen. And we think that we can learn something from bats about getting out ahead of infection and mitigating disease course early on to prevent the very more severe symptoms and the burden on the healthcare system. Obviously, lots of questions remain unanswered, a lot of the goals that Judith mentioned at the beginning, but we have established this experimental protocol now so we can begin addressing them. We know that we can actually do this kind of research. And to that end, no other groups that we know of that are investigating bats as reservoir hosts are using this particular approach, which we think will allow us, especially to uncover new and exciting things going forward. So I'll just end with our acknowledgements. Of course, Dakota, again, I mentioned our in-residence RNA-seq analysis expert. And then, of course, our collaborators. So Amiko, who's here at the Biodome in Montreal, she's actually the veterinarian there, and she's the one who taught us how to do the bat bleeds. Luis Barrero and Vanya, who helped with the simulations and taught me how to do that. Of course, Rolf and Weijing, without whom we couldn't have done literally none of this. Michelle Baker in Australia, and of course, Shaw Warren, the head of the Spirit Project. And this project in particular was funded by Judith's Canada Research Chairs grant as well as DARPA, but the lab has a variety of funding that supports us in our other work, in particular CIHR and CIRC and the Human Frontiers program grant. So I think with that, we can take any questions.